Hello, this is lesson 6 on cellular respiration for the macromolecules unit, part C for the assignment. In this video session we will be outlining the key stages which are part of aerobic respiration, explaining how ATP is produced. Now this comes with a warning, at which point you're all holding your heads in your hands, that this is probably the most difficult thing to teach and one of the most difficult things to learn, particularly in this assignment, because it only says outline the key stages. So if you're outlining something, you're only sort of giving a brief summary. Now it's really difficult to give a brief summary in this thing because it takes an insane amount of time to actually learn it properly, and there's a lot of biochemistry going on. So I'm going to have to go into more detail than I need, but hopefully it should make sense. You will need to go through it again and again because it will not go in first time. There are some fundamental takeaways that you desperately need to understand, and we'll hopefully build on those, and you can replay this again and again. I do recommend the Khan Academy videos to help support you with this. He breaks them down really nicely, but again, even those will be in far more detail than you ever need. So let's get to it. This is the GCSE version for a chemical equation for respiration. Okay, you can see on the left hand side there's glucose and oxygen, which is very simple. The stuff that we eat and the stuff that we breathe goes around our cells, around our blood should I say, to our cells. We make energy with it, which is on the end of there, and as a byproduct we create water and carbon dioxide. Now that fundamentally is a relatively simple process. I'm sure that we all understand that. The complicated bit comes in when we try and start talking about ATP. ATP, or adenosine triphosphate, is the energy currency for the body. When we've talked about processes like a protein pump, when we've talked about things like exocytosis, or anywhere that's needed energy, any active process, we've kind of seen this molecule, which is ATP. We have enough stored in our bodies to last probably, I don't know, between 10 and 20 seconds, so we constantly have to recycle it. And it's this breaking down of glucose and oxygen which allows us to create ATP. So we need sugar and we need oxygen to be able to recycle this ATP to help us stay alive. Now under optimal conditions, which means the best possible conditions, for every 1 molecule of glucose we can produce up to 38 molecules of ATP. That's just something to keep in mind when we move on. So a little bit about ATP to start with. ATP is this bottom molecule here. It's an adenosine molecule attached to 3 phosphates. Hence ATP. Adenosine tri, meaning 3 phosphates. Now that should be relatively straightforward. The molecule on top is a molecule called ADP, adenosine diphosphate, which means 1 molecule of adenosine and 2 molecules of phosphate, which you can see hopefully here. So in order to make ATP from ADP, what we've got to do is somehow attach another phosphate onto the end here. And that allows us to recycle ATP in a process called phosphorylation, basically adding a phosphor group onto the end. What's the big deal with ATP? Well, when you break this third bond, you get energy. So we spend a lot of time attaching this third phosphate onto the end of ADP, so that would be APP. We spend a lot of time attaching this extra phosphate group onto the end of ADP. Why? Because when it breaks down, when we actually break this bond down and free the phosphate, we release a lot of energy. And it's that energy there that drives the processes which I identified before: exocytosis, protein pumps, those little motor proteins that walk up and down. So that's the reason why ATP is so important. And if we sort of looked at ATP properly, what we would see is that it's actually a coiled spring almost. So when ATP is made or created, we get this kind of coiled effect, and there are bonds that hold it together. So when we break this third bond, this phosphate pops off. We've now got A and two P's and this free phosphate out. And what happens is this, once this bond breaks, this sort of spring action occurs. So it twang, it fires forward, and it's that produces the energy that the cell can use. So we end up with A, D, P and P. So what happens is that P has to get put back on somehow to make ATP again, and that means that it can be made and used to create energy in exactly the same way that I've just explained right there, clear as mud. So in order to make ATP, we go through a process called aerobic respiration, and it has 3 stages. Now some textbooks will say 4, and it's perfectly fine if you want to say 4. The first stage is glycolysis. The second stage, in some people's eyes, will be the link reaction, and that tends to be the most common one, but I'm trying to simplify it in this video. The third stage would be Krebs cycle, and the fourth stage, or third stage in my presentation, would be the electron transport chain. I've put these two together because it kind of simplifies it a little bit, if that's possible. So for 1 molecule of glucose, it goes through a series of reactions during these 3 processes, which end up in the creation of 38 ATP molecules. Now the question is, where does this happen? Where do we create ATP? Where do we create energy? Where are the energy factories inside our cell? The answer, from your own knowledge, should be the mitochondria in the cell. So let's have a look what goes on in the mitochondria. Before we do that, it's really important to familiarise yourself with the structure of mitochondria, or a mitochondrion. So this is the cytoplasm around the outside, the fluid of the cell, and you can see that it's kind of an inner matrix. There is cristae, which are these folding membranous parts, or internal membrane, and then there's an outer compartment, which is this grey area here. So you'll find that the 3 or 4 stages that we talked about occur in different parts of the mitochondria, which can be summarised here. So glycolysis, the first stage of aerobic respiration, occurs in the cytoplasm, not in the mitochondria at all. The second stage, Krebs cycle, occurs in the matrix, and then the electron transport chain occurs in the cristae of mitochondria, i.e. the folding membranes. So keep that in mind when you're learning about it. So the first stage is it goes from outside to the middle and then back a little bit, if you see what I mean. Right, now I guess into the more complicated bit. So we've got the 3 stages down this right hand side, and we've got the amount of ATPs produced at each stage. So glycolysis creates 4 ATP molecules, but has to use 2 of them. So it's like creating 4 or baking 4 buns for a birthday party, let's say, and eating 2 of them. What a lovely bun drawing. So if we scoff 2, we're left with a net gain of 2. So the cell gets a net gain of 2 ATP molecules in glycolysis. Krebs cycle is the next stage, creates another 2 ATP molecules. But hopefully you can see that the vast majority of ATP molecules are created in what's known as the electron transport chain. Now that's for every 1 molecule of glucose. So every molecule of glucose that goes through respiration, aerobic respiration, produces a sum total of 34 plus 2 plus 2, which gives us that magic figure of 38 ATP molecules. That is under ideal conditions. It's not every time, and it is unreliable to some degree. Now, what's really important in this is that these 2 stages, or the first 2 stages, glycolysis and Krebs cycle, also produce something called NADH and FADH. Nicotinamide adenine dinucleotide and flavin or flavin adenine dinucleotide. What a mouthful. As it says in the footer here, they are ingredients which are used in the electron transport chain to create the ATP. So this 2 NADHs here produced in glycolysis, this 8 NADHs here produced in Krebs, and 2 FADHs produced in Krebs, excuse me, are passed into the electron transport chain and end up producing the vast bulk of ATP in cellular respiration. So this is how you calculate the numbers. Each NADH produced creates 3 ATP molecules in the electron transport chain. So everything in this column here that I've just identified in purple, each NADH produces 3 ATP molecules. So we've got 2 and 8, which make 10. 10 NADHs go into the electron transport chain, and each one of those produces 3 ATP molecules. So you can see that these 10 NADHs produce 30 ATP molecules in the electron transport chain. The same goes for FADH. 2 molecules of those are produced in the Krebs cycle. Well, 2 molecules go into the electron transport chain, and each of those molecules produces 2 ATP molecules. So 2 times 2 is 4, and that's where you get your 34 from. This produces 4, this produces a total 30, so you get your 34 plus your 2 plus your 2 gives you a grand total of 38. That's the numbers involved. You may well have to repeat that little section again and again to try and figure out where the 38 ATP molecules come from in the total of cellular respiration. Hopefully that makes sense. Excuse me. So here's a bit of a pictorial view. So from glycolysis, what we've got here is the 2 NADHs passed into the electron transport chain, and here in Krebs cycle you've got 8 NADHs passed into the electron transport chain and 2 FADHs passed into the electron transport chain. That's where the magic happens. That's where your 34 ATP gets made, and that's where your 2, and that's where your 2. Grand total 38. Clear as crystal. Now you can pause this and have a look at it. It's exactly a summary of what I've just said. If it helps you, hopefully it will, just to sort of nail down the maths behind it, where each molecule of ATP is made and how these get passed into the electron transport chain to create ATP. Have a look and have a read. So this is the first stage of glycolysis. I know when you first see it you probably want to spew upon your piece of paper, but actually it's not too bad, and it's a very, very simple summary of what is a very, very complicated process. So if we look at the summary at the top, we said that glycolysis takes place in the cytoplasm of cells. Now, although it says it is anaerobic, and it is anaerobic, it's still required because the products, the pyruvate at the bottom, go into aerobic respiration. So we need to go through this before we actually start aerobic respiration, and we can't go into aerobic respiration without doing this. So it's required. The net production of ATP for this process is 2, but we actually produce 4 but lose 2, so we get a net gain of 2, and we also produce 2 NADHs with 0 FADHs. So firstly, let me summarize what's going on in this diagram. The green balls refer to the amount of carbon atoms in glucose. So glucose has 6 carbon atoms in the spine of the molecule. Now if you've taken in the structure of the carbohydrates video, you'll understand that. And what happens is that this is our energy investment phase, which you can see down the side here. We actually use 2 ATP molecules, and you can see that because it goes from ATP to ADP. So if you remember, there's the A, 1, 2, 3, ATP molecule. We use those to create energy. So where does the spare phosphates go? Now we've got 2 of these, so we'll have 2 free phosphates. Hopefully you can see they get attached. These 2 free phosphate groups get attached to either end of the glucose molecule. So here's what was glucose. Now it changes the name of glucose, but it doesn't matter. We don't need to know it at all. But all we're doing there is showing that we're using some energy to add on these 2 phosphates onto the molecule of glucose that entered. And what happens next is they split into 2 3-carbon molecules. 1, 2, 3, 4, 5 and 6. And if you follow the diagram down for each 3-carbon molecule, then another phosphate gets added. So we've got this one being attached, and we've also got this one being attached. Okay. So we've now got 2 3-carbon molecules with 2 phosphates attached at either end. So remember, we produce 2 NADHs in this process. So all that really happens is this NAD comes along and grabs essentially, how can I say it? It grabs electrons. It's probably the best way to put it. So it turns NAD into NADH. Remember, we're producing 2 NADHs in the whole per molecule of glucose. So this NADH here and this NADH here buzzes off to go and join the electron transport chain in the cristae, waiting to be used to create the 34 ATP molecules in the electron transport chain. Then finally, what we get is that 2 ADP molecules on this branch and 2 ADP molecules on this branch are converted each to 2 ATP. That's where we get our 4 ATP molecules. And it's just this phosphate here and this phosphate here gets attached to an ADP molecule. So this is happening times 2. So you've got, this is your spare one. They're just getting attached on this side, and the same thing happens on this side here. 2 ADP molecules get 2 of the phosphates on this side, and that's where your 4 ATP molecules. So we've spent 2 up here in our investment phase. We've gained 4, which means we have a sum net total of 2. Whew. So, in summary then, glycolysis, what we can do is simplify it to track the number of carbon atoms, the green balls. We can track the phosphates that are either attached or disattached from ADP and ATP. And we can also show that we're producing NADH from NAD. The final product that we get is these 2 3-carbon molecules called pyruvate. And pyruvate in anaerobic respiration, so if there's not oxygen present, if we are sprinting, let's say, and we're doing anaerobic respiration for 10-12 seconds, then that will produce pyruvic acid, and that's where we'll get lactic acid from almost. If we have oxygen present, then that goes through to aerobic respiration, and these 2 pyruvates you will see eventually enter the Krebs cycle. Before the Krebs cycle, this is the sort of stage where you see it added in some sources but not others and joined with the Krebs cycle. Before the Krebs cycle starts, we have to have something called the link reaction. It takes place in the mitochondrial matrix, which is the inner part of the mitochondria. So to summarise, it converts each pyruvate, so remember those 2 3-carbon molecules that we ended up with, into acetyl coenzyme A. That's not to be confused with acetylcholine in terms of the neurotransmitter in muscles. Acetyl coenzyme A in preparation for their entry into the Krebs cycle. Do not be fazed by just these posh, biochemistry names. I don't know who came up with them, but they sound very posh and very confusing, but it is a very complicated topic. So what happens in the link reaction? Well, these are the 2 pyruvates that came through from glycolysis at the end. And all you can see there is on each we produce some carbon dioxide. Now notice carbon dioxide has an atom of carbon in each. So it turns a 3 carbon molecule into a 2 carbon molecule. And that is the carbon dioxide that we're breathing out right now. That's a byproduct of respiration. And you always know that, red blood cells drop oxygen off, they pick up the carbon dioxide. This is where it's been produced. It's been produced in the link reaction in the mitochondria. What do we get? We also get 2 NADHs being produced. So basically, they grab a hydrogen atom, and we end up with acetyl-CoA, which is a 2 carbon molecule. Now don't forget there's 2 of these per glucose molecule, because the 6 carbon glucose molecule split into 2, and for every glucose molecule this happens once and twice. So 2 things go into 2 Krebs cycles. Alright, so the cycle that I'm just about to show you, this happens twice for every glucose molecule that comes down the pathway, let's say. Okay, I'll let you read that in your own time. And there's a summary of the products produced by Krebs and by the link reaction. Okay, so as a result of the link reaction, we get sent through a molecule, a 2 carbon molecule of acetyl-CoA. Now don't forget that happens twice, so there is another Krebs cycle going on somewhere in the mitochondria for every molecule of glucose. So this is happening twice. And at the start of the Krebs cycle, it mixes with oxaloacetate, which is a 4 carbon molecule. So 4 carbon plus 2 carbon gives us a molecule of 6 carbon. Okay, so we've got 4, 2, which gives us a 6 carbon molecule. Now you can see the next molecule is a 5 carbon, and you will not be forgiven for noticing that we produce more carbon dioxide. There's the atom of carbon. We're going from a 6 to a 5 carbon compound, and we lose another one to go back to this 4 carbon, and you can see hopefully that it's a cycle. So this 4 adds with a 2 to give a 6, which is here. We lose a carbon, which takes it down to a 5. We lose another carbon, which takes it down to a 4, and it just keeps going around in a cycle. And that's why it's called the Krebs cycle, sometimes called the citric acid cycle, because obviously the 4 plus the 2 gives us citrate, which is citric acid. Okay, so let's have a look at the ingredients that are being included in this citric acid cycle. So hopefully you can see, let's just track. It should match up with the figures that we put on one of the first slides in the amount of NADH produced. So here we've got 1, here we've got 2 and 3 NADHs. Now don't forget that's happening twice, so that will be 6 plus the 2 in the link reaction would be 8. We've also got an ATP molecule being made here, so that would obviously happen twice, and that will give us the 2 ATP molecules, and we've got 1 FADH molecule, which would give us overall, if it's happening twice, 2 FADH molecules. So if you go back to the initial slide where it summarizes exactly our ingredients, you can rewind it, then you should be able to check that these ingredients here are accurate, and these NADHs are the things that goes to the electron transport chain. These are the ATP molecules that go for the cell's energy requirements. We've got CO2 that we're breathing out, and we've got the FADHs again going to the electron transport chain. So I thought this would be a nice image or video to play, but I'm not sure whether it's going to record it. So if it doesn't, then it doesn't. I'll upload the PowerPoint to Google Classroom, but what you see in there are proteins on the cristae, and this is where the electron transport chain happens. Will it play? It will. Fantastic. And what happens here, this is where the 34 ATP are being made. They're protein turbines on the cell membrane spinning around fast, attaching the third phosphate onto ADP to give us a 34 ATP. That's what I'm going to explain in a second. So this is it, the pièce de résistance. If you remember, the last two stages have been all pretty much geared up to passing NADH and FADH through into the electron transport chain. And you'll have to go back to figure out or to remember how many, I've forgotten exactly how many NADH come through an FADH, but they essentially make 34 ATP in this stage. And what happens in this stage is the hydrogen off each NADH and FADH disattaches, and if you remember our lesson on atomic structure in terms of hydrogen, a hydrogen atom consists of one electron around the valence shell and one proton. That's all it's got in it. So the proton, which has a positive charge, pops off, and the electron with a negative charge pops off. Now the electron does a load of fancy stuff with energy. We're not going to focus on that because we need to simplify it. The protons are effectively pumped out. We know it's pumped out because we're using energy to do it. So these protons are pumped out from the matrix to the outer compartment. Now at this point, stop the video, go back to the structure of mitochondria, and it'll tell you where it is. So if you remember, there's a mitochondria, there's a cristae. So essentially they're pumped from the matrix to the outer compartment. That's where they're going. They're going out there, or out there, or out there. These protons build up a positive charge, and they go into this, I don't know, this turbine called ATP synthase. And as these protons rush through, it spins this turbine at a ridiculous speed. So go back to the last animation that I just showed you. It finished very quickly, but as you can see, they spin. And as they spin, what happens is they attach free phosphate onto ADP. So these free phosphates go onto the enzyme. As the enzyme spins, the energy used from that adds on the third phosphate, and what comes off is ATP. And that is how ATP is produced. I can't wait for all the questions, but rewind it and come and ask me.